one of those sponges in the background that looks like it's either been knocked over.
Half
Maybe
gone.
some of it's still
Zoom in
living.
on that?
Some parts of it might be living, or if it recently fell over.
Mm-hmm.
Looks like there's definitely some other creatures that are using it as substrate to live in and on and around.
Maybe there's a scale worm, that cute little pink purple thing to the right of the sponge.
Oh, I see that. Look at that. And then there's one on the sponge.
Oh, yeah.
Is this pieces of crab leg or something?
Ooh.
Looks like
Oh.
someone was unlucky.
And what's that? Oh, that's
Can you
a
zoom
worm.
in more?
Looks like it.
Some sort of cool.
Somebody
spaghetti worm to the lower left with the tentacles coming out.
got munched on there.
Oh, yeah, there's the scale worm.
2 different scale worms.
Ooh, one on the sponge and one to the right. Very cool zoom.
And I see another spaghetti worm tentacle.
Yeah.
Oh,
Yeah.
cool.
And do you think that that's his last meal, that spaghetti worms? Those little legs, what did they prey on?
Spaghetti worms just eat the detritus. They wouldn't be able to knock off a crab like that.
It just molted?
Maybe that was a predator.
Yeah, something just went by on the right. All we see is the shadow.
Yeah.
It's a beautiful image of that scale worm.
Or maybe there's a creature inside the sponge.
The way the pieces are broken up, I'd say someone's had a meal.
Yeah. That crab is.
That crab.
I think we'll have to get on our way.
Guilty. Look at that guilty look on his face.
Hey, zoom out.
Zooming out.
Bye, scale worms.
Junk zoom.
Audio check. This is Megan on V-Link.
Hi, Megan.
Hi, Megan.
Hi, Megan.
Hey, Megan.
Hey, everybody.
Greetings.
So we're going to do a little special Q&A while you're on the slope here. Thought it might be a good time to answer some audience questions.
Sounds good.
Got an overhang there.
Yeah.
Ooh,
Wow.
Steep
cool
here.
Yeah.
little rock creature.
Sorry, Rennie was getting nervous.
I know, I kept looking over. I was like, gotta get up that thing.
Ooh.
Ooh.
Whoa.
Oh.
Mating
Or predation. Who knows?
Oh, no.
That's West Side Story there.
Look at that.
zoom in?
Coming.
Why are they
Oh.
different colors?
Whoa, that's a tight grip.
That's
Yeah.
face to face.
They're either having a disagreement or are really in love.
Or somewhere in between.
I think they're color morphs of the same crab.
Whoa, this is incredible.
It's a kind of king crab, a lithoted crab.
You're just kind of gently
Can
touching.
you,
It
can
could-
you give me a stick lock?
Yeah.
What do you guys think? Mating or predation?
Oh, we might find out here. Wow.
Well, Paul was telling the
Big
story
deal.
the other day when mating can happen only when molting has happened in some crabs. And often the male will grab hold of the female until she molts, so no other male can get access to her. So, not sure if that happens in king crabs.
Hmm.
They're like flicking their little central antennae.
Yeah,
Yeah.
I think it could be a pre-nuptial, let's call it courtship.
Man.
You gotta get
Wall's
moving.
getting close.
Okay, moving on.
Might be a long wait to see anything more than this.
We're gonna be seeing the rock up close and personal. Hang on too long.
Entire rock here.
Yeah, the ship just came to a stop, so we'll get a little bit of time to catch up and before I call in another one.
That's a
There's a scallop down there.
Lower left.
Can we zoom in on that? I
Is
think
that a
that's,
Acesta?
it could be a cesta, which would be good to collect.
Ooh, it was open when we came upon it, right?
Yeah. Just closed
This is a
up.
kind of clamp.
Ooh,
Cool.
be camouflage there.
A landslide?
Oh no.
I was hoping to see these on the San Juan seamount. We saw one, and they were very delicate shell, but it's on a rock, so we might be...
We have a visitor over there.
I think it's
Yeah.
just a stalked sponge that's creeping onto the porch.
Could actually be a scallop and not an Acesta.
Hmm.
We'll see the hinge in a minute.
Hey, zoom in.
Coming in.
Oh no, that's Acesta.
Oh, cool.
Wow.
So it was open.
Acesta.
Great eyes spotting that.
Maybe we make a rock collection
Yeah, let's
with a bivalve.
Yeah,
let's take that,
that'd be really cool.
please.
Jake, we're pretty stable.
Very good.
Yeah, I think.
Okay.
Good to
Let's
get.
see if we. I need to back up maybe.
You can
Let's back up a little bit.
just barely on the edge of down cam. You got him.
Yeah, so maybe you can turn around and get a view.
Yeah.
We're
Well,
stable, so I don't think we're going to hit anything.
Okay,
Like-
zoom out.
Coming out.
Yeah, Bob, if you can get it with the rock, it'll be the biggest biology Lisa's ever recorded on a single rock.
Wow, you might
Megafauna.
be bigger than the rock.
It might
Yeah,
be.
Megafauna, yeah.
It might be tugging Herc's tail there
Oh,
for you
yeah.
to turn around. So.
The bio to rock ratio is pretty much 1 to 1.
Yeah, don't yank me off.
Come down a bit too, I guess. I have a little bit of room.
Yeah. Yeah. Just watch that too.
That's
Yep.
pretty cool. Ooh.
You could see it in your main HD as well.
Yeah.
Okay, zoom back in.
It's pretty small. Maybe we'll just put it in the general collection box.
Fortunately, those are full right now.
We zoom in.
Yeah.
Coming
Oh, they've got samples in them.
Yeah.
Got it.
There's a hagfish in the background.
Put it.
No, okay.
No, we can just put it in between.
Yeah, let's put it in A or B. Yeah.
Stranger Things reference there.
For those of you watching at home, the ROV hand is
Bigger
about
rock.
the same size as a human hand.
Bigger rock than
Very
anticipated.
cool.
The iceberg rock.
It's a quartz.
Interesting white. Yeah, I don't think Kira
Yeah.
will be interested in it for geo, so we'll definitely take it.
That's like a sponge or something on it, right?
Is that, or is it quartz?
Wanna
Looks like-
zoom in?
Or,
coming in tight.
or is it carbonate?
That
It
looks
does look like
sponge.
quartz.
It does.
That's not quartz. That's sponge or something, right?
Hard to say.
Oh.
Only science.
It would be
can
quite
tell.
altered on top if it was.
Do we expect there to be quartz out here?
No idea. One of our onshore geologists,
Oh,
Kira, could
Crispin saying quartz.
chime in.
Wow.
Oh,
Yeah, that looks.
sorry, I
rock-like,
thought you
yeah.
guys could hear me.
Oh, what do you think, Kira?
It's definitely not the minerals of interest.
No, we're not going to put it in a collection box. We're going to keep it as a bio sample in
Oh.
the.
I think it's likely a terrestrial rock. You don't get quartz in the deep ocean.
Yeah.
So yeah, whatever you guys want to do with it, I can take a look
What-
at it.
Yeah, we're taking it for the fauna, but not quantitative for Lisa.
Okay.
Thanks.
Okay, zoom out. Starboard box.
Yes, please.
Yeah, and if we could put it in the forward, kind of in between space.
Do you think it'll fit?
Yeah.
That's the
10
only space
centimeters
we
at least.
have, Yeah.
Let me see the end of the arm there.
You have another place, Greg, we could possibly put it?
Porch is the only other place.
Yeah, we've got where the one bone basket was.
Well, if we don't think it'll fit, then we can put it on the porch.
I think you'll lose the cesta
It might. It's pretty delicately attached.
if you have it on the porch.
Hopefully. Nice.
So they're aiming for one of the gaps, Bob.
Yeah.
Yeah.
Forward gap.
Or you could knock the clam off into the gap. Lisa doesn't want it in any of those chambers, though.
Gap.
It won't be attached. It's just held on by a few threads.
Yeah, I think the rock's kind of big to get
Too big. Yeah.
in the gap. We could knock the clam off.
If you can try and knock it off, that would be great.
And what's it going to knock off into?
One of the gaps.
Or
So
one of
the
the gaps.
forward
In a gap, yes,
Yeah.
please.
There's the forward and kind of more aft gap between the boxes.
You didn't think we were going to make your life easy with this, did you?
Oh,
There you go.
down in there?
Yeah.
Perfect.
I believe that's going to be the aft
The B gap.
B gap.
Maybe.
Maybe.
Wow.
Looking promising.
Oh,
there?
nicely done.
Yeah.
Beautiful.
Oh.
That's going to be
We're not keeping the rock then. We're
No thanks.
pitching.
sample
huh?
NA124.
Pitch it.
It's a really nice
157.
rock.
Unless Kira wants.
Yeah. Nice
Unless
quartz
Kira
rock.
would like.
Souvenir rock.
Well, we could put it on the porch.
Sorry, that was 157?
Yes,
I could use the
157.
weight, really.
If you want it, Bob, you can take it.
I know Jess would take it. We'll just throw it over here then. What's that?
Under Mongo.
And should we call this the same sample? Would you like the same sample number or a new sample number on this rock?
The rock's just ballast.
Yeah, ballast rock.
Give it a number.
Give it a number?
Call it R. Is it R4?
Yes, R4, and that's going to be sample number NA124158.
Okay.
Rock number 4.
Eight.
And it's stored on the porch.
You know, Bob, while we're here,
Yep.
we have an opportunity to collect one sediment sample via suction.
Huh. Okay.
And so I wouldn't mind getting that while we're here.
And that's a jar 1?
That's correct.
Okay. Jar 1.
We kept the rock mark. It's it'll come up.
What will you be looking at in the sediment sample and wanting to examine?
Where are we going? Just any old place in here?
Yeah, good question. So generally yeah, anywhere in there. Just
So it looks like there's some stuff in jar 1. I don't know.
Some remnants.
stuff.
Just remnants.
Can we
Yeah.
blow
You get
that
around the
out, or
flush.
can you
Do a
flush
flush?
it, please? Yeah, thank you.
Flush it. Just run it.
All right. Running.
Yeah, so generally we use the push cores to collect sediments, which gives us a very nice way to collect them undisturbed. And we can then section them, and we're
Although it is the fine mesh.
particularly interested in the bacteria
Yeah.
that live in those
I don't think it's going
sediments.
to go out.
Yeah.
Lisa and others are interested in the
All right.
invertebrates that live in them,
Suck it
so
up.
we have some different goals with our sediment collections.
It's kind of clear.
All of it relates to
Yeah, it looks good now.
documenting biodiversity. And so this is not a traditional way to collect sediments for us, but nonetheless it is a way to try to capture some of these surface sediments. We put
You need
a
to zoom in.
fine mesh screen on this,
I want to collect
the
a
end
bunch
of
rocks.
the jar
Get them out of the
that the sediment's being sucked in to try to capture it.
Yeah.
And what we're going to do is ask questions about what types of bacteria are living in these sediments. And then also we're going to assess their potential to make these things we call natural products, or small molecules that have historically been important for us as a source of medicines like antibiotics and anti-cancer agents and other types of drugs that have been so useful to help treating disease. So people have not really studied the microbes that live in the ocean for this purpose. We, for many years, have been studying microbes that live in ocean sediments and found that they can be very different than the ones that live on land. I guess that might not be a complete surprise. And we've also found that some of these microbes can make molecules that have never been seen before. So we're very excited about the prospects of getting a better understanding of the microbial diversity in these habitats, and also their potential to produce these molecules. And so we're calling this biopharmaceutical potential on this trip. And we're studying that both in the microbes that live in these sediments, and we're also collecting certain vertebrates that are also known to produce these kinds of molecules. And generally, when they do, they're for defensive purposes. So things are very different here. We're more used to working on coral reefs where there's a very high density of organisms, and basically everything you see is either has a physical defense or a chemical defense. And so we just collected a bivalve. There's a perfect example of something with a physical and structural defense. And interestingly, the things with structural defenses are often the things we like to eat. Bivalves, clams, scallops, lobsters, shrimps, and sea urchins, right? So the things that don't have obvious structural defenses, at least on a coral reef, will often be chemically defended. And those are not often things that we're known to like to eat either. So we don't know as much about the role of chemical defense in these deep ocean environments where I suspect predation is not as great as on a coral reef.
Do you think we have enough?
Yes, I think that's looking great. Well done, thank you.
And just to confirm, this is sample NA124 159.
Yep. 159.
Yes.
Roger.
That was a great explanation. Thank you.
Thanks for the question, Megan.
Caitlin,
Yes.
could you write to Steve Alskovich, just ask him about the remote capture further? The still camera seems to have frozen, at least the feed I'm seeing.
It's just the remote desktop connection.
It's okay?
Yeah,
okay.
I can bring it back up.
No worries. Thanks, Jake.
Following up on that question, how long will it take you to get through all of those processes? Like how many months of work did we just vacuum up for you?
Or did you mean years?
Okay.
Yeah, well, that all depends.
Bob.
And so
Scooting over.
we're hoping to get some preliminary understanding of what bacteria live in these ocean sediments and within the next couple of months. But we could be studying these samples for years.
Interesting rock wall there.
Yeah, it's also interesting that there's no coral on it, but maybe that has to do with the oxygen?
Are these the kind of rocks that Kira was interested in, or are they too angular?
That looks pretty basalty, right?
Yeah, Crispin Little is actually chiming in on the science chat, saying that these outcrop patterns here don't look like extrusive basalts, no pillows or columnar jointing.
Columnar, yeah.
But neither do they look like sediment, so no obvious bedding. So they're obviously hard. And Crispin's thinking that they could be some type of metamorphic rock here. It's definitely a little bit of the different structure that we've been seeing.
Does anyone want to take a stab at explaining why it's tilted that way?
Sorry, I didn't catch that.
It's sort of tilted at interesting angles. Anyone want to hazard a guess why that is?
Oh, you're asking biologists a geology question?
we have an expert who has been chiming in on the science chat, and Crispin Little just sent another message.
We're gonna call a friend.
Uplift.
You might want to adjust your mic to be closer.
Uplift.
So Crispin has come back saying that there does seem to be some type of layering here, and so perhaps some of these could be sedimentary. Although Crispin's also noting that layering does happen in metamorphic rocks.
Looks like a sort of old one.
Yeah.
Right.
Sure, someone out there can date that can based off that design.
Got our Pepsi specialist in the science chat.
I bet we do.
It's 1972.
Yeah. Well, it's got a hole worked in it. It's been there a while.
Coming out.
I gotta get moving here anyway.
So Mark Legg in the science chat is saying that rhyolites, dacites, I'm probably completely butchering these names, and ash flow deposits called tuffs can have this blocky appearance, and cherts and shales, diatomites, etc. in the Monterey area are also hard and fractured. So perhaps some of those could be what we're seeing. And Mark Kammerling is saying, giving us some more information, saying that it could be a slide block, which could cause tilting, or it could be structural associated with nearby faulting. So perhaps, Megan, that's getting to your question about asking us why these might be tilted. So thanks so much for all the scientists ashore who are sending us messages in the science chat.
Pretty cool looking from this,
There's
angle.
There's additional depth to that. Look at that.
Our phone a friend worked.
That's
We
perfect.
just ask our chat. It's great.
And viewers, keep your questions coming in. We'll answer questions for about an hour here. And that was the 1987 to 1991 Diet Pepsi.
Wow.
Thanks, Cook. Appreciate it.
Still looked fresh.
Very square looking sponge over there on the left.
Want to zoom in?
Out on a little perch.
Yeah, perched on another sponge, it looked like a dead sponge.
With a Panacea sea cucumber in the background.
My niece Marina just texted me and said, listening, watching to your live feed. Pretty damn cool! Exclamation, exclamation. Hi, Marina. Glad you could join in.
Okay, zoom out.
We have a question from a 3rd grade teacher who's tuned in watching with us. One of their students who's watching would like to know what the most unusual animal is to research. I have a bunch of biologists in the room. What is unusual to you?
There's so many. Good question.
We all look to the invertebrate zoologist.
I would say this is Kyla Pearson speaking, and in the deep sea we find so many unusual creatures. I mean, the list just goes on and on and on. I think what brings credence to their unusualness is their adaptations. I am studying a type of deep sea worm that is a parasite to another type of worm, and for me, I think what's super cool is how these animals have evolved to withstand these crazy extreme conditions. And my worm is actually in the process of losing its eyes because there's no need for eyes when there's no light. So I would say that the worm that I'm studying, a type of phylodocid, is pretty dang unusual.
It was a really solid choice. Anyone else have a favorite, most unusual animal?
Well, Renee and I were talking last night about his favorite worm, which is the flamboyant squid worm.
Yeah.
And Nautilus collected one in 2016, was it?
2015.
2015,
yeah.
off Galapagos.
Yeah.
But it turned out we'd actually found the original flamboyant scale worm off the Philippines and named it in 2010. And why it's fascinating is that it looks, and when the people first saw it, they thought it was a cross between a squid and a worm, and that's why they gave it the nickname squid worm, because it had 10 appendages, a bit like a squid, 10 tentacles. But in fact, that was 8 gills and 2 tentacles for feeding, and it really was a worm related to earthworms, but it was swimming beautifully. We've studied its evolution, and it turns out its ancestors were living on the sea floor.
You moving?
And it evolved to become a swimming form, and we've got new observations on it, and it seems to dive down to the bottom and touch the sediment and scoop it up with its tentacles, and it still lives the way its sea floor dwelling relatives do, in that it eats mud, but it has this beautiful pelagic lifestyle otherwise. So if you look up flamboyant squidworm, you'll see some wonderful Nautilus video online. So it's mine, one of my favorites as well as Renee's.
Yeah, it was actually pretty, it was pretty incredible. We found one and then sampled it, and then later on, I think it was a couple dives later, we were looking for a vent site. It was pretty deep, so we're just kind of scouring around for it. And we were zoomed in on this other flamboyant squidworm in the water column and kind of had just our focus on this, and it was right in front of our faces. And then we're like, okay, that's enough of that. And as soon as we changed the focus to the background, it was this vent structure we were looking for. So it was, the squidworm led us right to it.
So what do you think, Greg? You want to make a beeline for our deployment site, or?
Yeah, I think this hasn't yielded us a lot. Renee, maybe we'll replan and just go up to AJ.
Sure, yeah, we can go up slow. Just head right
and then we'll have a rethink after that. Maybe on the way up we'll see something different as oxygen changes.
Sounds good. All right, Jake, we're going to switch to 085.
085,
so just going to kind of head upslope.
Okay.
So for those of you tuning in, we've got quite a few objectives left in this dive, including deploying some experiments that Greg and Lisa have designed, and Greg will probably tell you all about them when we get to that point. We're also hoping to make some more collections, in particular some soft corals and maybe a sponge or two.
Yep.
So
it's gonna be
our 4-hour shift goes very quickly.
So we also have a few more transects to do.
Yeah, we've been going along at a similar depth now for a while, hoping we'd come across some filter feeders, different kinds of corals, but we really haven't. So we're now going to go upslope a little bit. Maybe we'll probably see the oxygen continue to decline, but we may still see a change in the animals as we go up. So we're going to head up.
Start a third transect here?
We could, yeah, probably time for another transect. We'll go up another 100 meters or so, and maybe we'll start to see some different kinds of creatures.
Can you handle that, Rennie? I know, I see you lost your pilot.
I'm running both. Roger.
We had some questions about how you decide what to do with all of these objectives. Do you want to talk a little bit about, within only the hours you have, how do you balance all of these different science priorities?
Oh boy, yeah. Well, amongst the science team, we have a lot of different objectives. The graduate students often have very focused projects, and different people will take different parts of what we're doing out here and work on it. Greg, for example, is able to fill in collections for many different projects he's working on. Some of what we're collecting will be
too many things
worked on by people who aren't out with us. We're a little bit limited in how many scientists we could bring on this trip. In part, that's due to COVID restrictions, and also just the nature of the operation of this ship, which requires a lot of personnel to maintain the ROV and do all the video work and all the communications. And there's a lot of data, what we call data logging going on, mapping the seafloor. Most of that happens in the evening when we're transiting from one site to another. So yeah, so there's a massive amount of information collected on these trips that then gets divvied up into a lot of directions. And a lot of it, there's a huge emphasis now to make data publicly available. And so there are a lot of scientists who aren't formally a part of this cruise that are participating from shore and benefiting from the observations that are being made. In some cases, they're asking us to make specific collections for them. And so it's really pretty remarkable how, with the technology available today, for those of you watching from home, and able to share what we're doing with so many people. But yeah, these samples will be worked on for many years to come. There's no question about that.
But in terms of how we work a dive, if you could see where we're sitting, we have a tally sheet of what we've done. We have a task sheet of what we need to achieve. We have a map of the containers that Hercules has where our samples will go. And those are push cores, boxes, containers that'll be sealed and kept cold on the way up. We have seven containers where the vacuum hose, the slurp, can suck into. And we would like to fill all of those. Our chief scientist, Lisa Levin, does an amazing job of doing all her research work as part of the science team, but also leading the planning for where we're going to dive and coming up with the dive plan for each day, and then leading the first half of the dive. And as we go along, we keep in mind the core priorities, which are filling the samples Some are more important than others. We've planned these dives so that normally we start on sediment a bit deeper and we get the push cores all done. And then we know we need to collect 5 different rocks. Ideally, they'll be encrusted rocks with ferromanganese or phosphorite and with their animals, and they'll be isolated in separate containers. And then we want to do 7 different slurps along the way, as well as pick up corals or sponges or other things that Paul and his team might want to look for active compounds, and they'll get put in the submersible slots as well. So we really have a core set of priorities, but then every now and then something strange happens, an opportunity to sample something. So we hope we have time to take a bit of time to, if we find, the other day we found a skeleton, or yesterday we found a piece of kelp on the seafloor. So we can spend half an hour often looking at those things. But then we need to make sure that we can pick up and get the objectives. We don't want to miss a rock that we wanted to collect, or miss push cores, because those are the core objectives for a particular dive. So time management's an issue, but that's why we have a team of people back here and a great team of pilots, navigators, and videographers ahead of us.
Perfect, thanks. Lots of questions coming in. Again, here's just a great chance to get your questions in while we're here, so go ahead and send them in. Our 3rd grade class is tuned in. I like this question a lot. They're doing research on the different kingdoms of animals, and they want to know if there are funguses in the ocean. I don't know the answer to this. What do you all think? What do you know?
Oh yeah, there are fungi in the ocean, absolutely. Many of them are in the form of yeasts or single-celled fungi. So if you collect a water sample, you will almost certainly find these single-celled yeasts in the water column. Much less is known about fungi in the deep ocean. In shallow waters, they play a big role in breaking down organic material, particular plant material. So if you were to go into, say, a mangrove area and collect some leaves from the mangrove trees that had fallen into the water, you would certainly find fungi helping to decompose those leaves. But interestingly, Greg is going to be part of the experiments they're putting out will include some wooden blocks, because they're interested in certain bivalves that are actually called worms, shipworms, but they're mollusks that colonize wood in the ocean, and we're going to take advantage of that. We were talking about that just last night, because we'd be very interested in finding out what types of fungi might colonize these wooden substrates that are placed deep in the ocean. And we're going to have a chance to come back here a year from now on a different ship, unfortunately, but nonetheless, we'll be able to come back on a different ship a year from now with a different submersible, and the plan is to recollect these experiments that are put out. And we're very interested in seeing what types of fungi might be colonizing these wooden blocks. And then again, all in the theme of biopharmaceutical potential, we will study those fungi. I think many people know that penicillin came from a fungus, right? Important antibiotic. So we're hoping we can find some new antibiotics and other types of molecules. Our primary goal is bacteria, but when an opportunity arises, why not take advantage of it? And we think these wooden blocks Greg's going to be deploying will give us a nice opportunity to see what sorts of marine fungi we can capture through these baits. So that's a great question.
Was that a
Trash
piece of
fish.
plastic plastic
Trash bag.
trash observation.
Yeah, so as you heard there, some expeditions are planned years out. Often the funding cycle for these is involved in multi-year processes where Ocean Exploration Trust secures partnerships to conduct expeditions. Many of the expeditions are planned sort of in that 6-month out time frame. Right now, the Ocean Exploration Trust team is working on the 2021 expedition schedule and starting to line things up while we're still underway on this expedition. And then this is the 7th expedition of the 2020 season. And if you want to check out all of those, click the expedition tab on the website and you can see where we've been so far this year and certainly back in history as well. Team in the van, you are a few days into the expedition, and one of the questions that's come in a few times here is, what is the most exciting thing you've seen on this particular expedition? So in the last, I don't know, 4 or so days, what has stuck out to you as particularly exciting so far?
Just right before we answer that question, I'm just going to go ahead and say it's the end of the transect.
End of the transect, thank you.
Got it.
Sure.
Well, we've got a couple of first-timer scientists party here with us.
I'll sit over here.
Kyla and Caitlin, so maybe we'll let them answer.
I'll go first. Kyla here. So far, everything has just been absolutely mind-blowing, and I feel so honored to sit in this seat with a few of the most famous deep sea biologists in the world, and we've seen some super unusual and super cool things. So one thing that sticks out in my brain was seeing the deep sea nudibranch. I love nudibranchs. They're so diverse. Their adaptations are just so cool. And yeah, I think seeing a deep sea nudibranch was definitely a highlight for me.
This is Caitlin speaking, and I'm going to have to 100% agree with Kyla. I think one of the most exciting things I've really enjoyed, so most of the day when we get our samples back, I'm a microbiologist, so I'm working with Dr. Paul Jensen and a postdoctoral researcher also on our team here on the ship, Gabriel. We're mostly processing the sediments and the different animals looking for symbionts and really interested in their bacteria. And so most of the night is spent sifting through and bagging up collections of mud. So one of the most exciting things during the day when we're doing these video transects and exploring during the 4 hours when we're watching what we're seeing on the ROV, I've absolutely loved seeing all the different corals, all the different brittle stars, all the different sponges that we happen upon. Some of my favorite creatures we've seen so far are the cephalopods or the octopuses. Those have been really, really cool findings, kind of creeping up on them, seeing where they're living. But we've seen some really beautiful diversity of creatures and colorful corals and colorful sponges. And I think that's been really exciting to see.
Very good choices. Sort of a parallel question. Somebody asked, what advice would you give, since it's your first time on an expedition like this? It might be a great question for you too, but what kind of advice would you give to somebody who would like to have the opportunity to do this, is interested in studying the same thing?
That's a great question. This is Kyla again. I would say to work really hard in school, get good grades, and most importantly, take advantage of opportunity. Get yourself out there, try and meet with people who have the careers that you want to have, and yeah, put yourself out there.
Just jumping onto that, this is Caitlin speaking. I would say anyone at any age, any walk of life, I think the most important thing is to stay curious, always ask questions and learn more about the world that we live in, the environment, the nature, explore things that are interesting and pursue those passions. And perhaps some people on deck with us who could speak more about the opportunities on Nautilus or how they became engineers and oceanographers on this ship would be Jake and Rennie. You guys, some opportunities specifically related to Nautilus and how you got your positions here?
I think we both started through the intern program, the OET intern program on the Nautilus. And I don't know, what position did you start in, Rennie?
I came on as a seafloor mapping specialist. My background during my graduate degree was in remote sensing. I was working with land-based remote sensing techniques like LiDAR, and then I kind of transitioned to, hey, here's an internship working with multi-beam sonar. How can I get into that? And that's how I started on the ship, and then have been in many of the roles. I was working in the wet lab for a while, also doing the mapping coordination and also ROV navigation, deep, like subsea nav. So kind of really it was all about that opportunity.
Trash.
Is that trash?
Trash?
Yeah.
Just that I would echo those sentiments, is that really be opportunistic. If you're laser focused on a very specific thing, you may miss some of the other opportunities that surround it that'll help you get there in the long run. So kind of be opportunistic and seek out those opportunities that'll help you get along your way.
I think I'm going to close that out with a quote that Jane Goodall's mother told Jane. And this is one of my favorite quotes ever. If you really want something, you have to work really hard, take advantage of opportunities, and above all, never give up.
That was a good one.
Here, here.
Absolutely. And if you are interested in the internships that Jake and Rennie mentioned, you can check out the education page or the team page where you can see the bios of everyone who's on board right now, but also learn more about those pathways onto the ship. We have paid internships in 5 different disciplines aboard Nautilus. And although our interns were deferred for this season, we look forward to bringing those programs back in the future.
And the food on board's really good.
Yeah, that's something about this cruise actually that has been nice is for the most of the season. We haven't had a lot of the science team, maybe one or two per cruise, or sometimes 0, and just solely, well, it's actually been great to work on shore with those scientists and kind of seamlessly have them, act as watch leads and chief scientists from shore. It really is nice, kind of missing out on some of having the excited scientist on board. So this cruise has been great for that, which is more typically how we operate with a lot of new people, either interns or people fresh in their graduate programs, and well-established scientists who have specific goals. It's nice to have that environment on board.
Oh, a nice little mushroom coral there.
Yeah.
First one we've seen of those, I think, maybe. First one I've seen.
I did see a while back, I saw another one of those blind lobsters.
Yes, Crispin also had noted that. That was a couple, 4 minutes ago now.
Yeah.
But nice spotting.
Do a zoom there, Jonathan.
Is that a rock pen next to it?
Yeah.
Anything of interest for you here?
Well, we could slurp some of those. We've got a rock pen. there on the right.
Gonna hang on here for a second.
You want to hold?
Yes.
Hold for a second.
Yes, we're just having a chat.
We're strategizing because I wouldn't mind
We could do some
another
slurping for the crabs. Yeah.
another collection of the mushroom coral, and then we can slurp for Greg. Please.
It's a really nice shot of that.
Would you like me to go to wide?
Yeah, you can come wide so I can settle in. Thanks.
Those are one of my favorites.
Bit of an Alice in Wonderland.
Off the coral slurpable.
Yeah, I'm backing up. I held position and we're backing up, but we may need to rise up and come back to this piece.
Okay.
In fact, yeah, I'd say so. So I'll just drop a, okay. Yeah, it's just I did back up, but it might swing a little bit. So we'll see how well that's arrested.
There comes that wall.
That's okay, I think we're all right. That's getting close. Yeah, maybe we should come up. Yeah.
It's looking pretty clear.
I don't know. It looks like 7 meters in the Argus sonar. If you guys want any push core samples, we can get one on Argus in a minute. No, we're okay, yeah.
So those of you not familiar with the operation, there's basically two ROVs. The Hercules is attached to the Argus.
I stopped and we're moving backwards.
Moving backwards.
The Argus is attached to the ship. So we have pilots for both. And this type of system allows Hercules
I'm sitting down.
Operate and maneuver quite freely without being pulled with the ship as the ship goes up and down with the seas and gets blown around with the wind. So it's a fairly complex system, but it allows the pilots to have really great control over how they position the Hercules.
Slurpos.
Yeah, that's how we get that super smooth video from Hercules flying around. It's not being yanked around by the movements of the ship or anything like that. Argus kind of absorbs all that. But it has limitations. Just have to be aware of where Argus is penduluming.
Great explanation, team. You're hired as science communication fellows. There's some questions coming in about this particular coral. Does anyone want to tell us some about mushroom corals or about this one in particular?
You hit the stick lock for me?
Yeah.
Thanks.
Stick lock. This is a colony. The ball that you can see there on top of the stalk gives rise to a series of different polyps, and those are the cylindrical structures coming out. And when we zoom in, you can look at some of those polyps. They'll probably retract pretty quickly once it gets a hint that it's going to be collected by the manipulator, and they'll pull in. But while they're still out, you could likely be able to count that they have 8 tentacles on each polyp, and that tells us that it's in a group called Octocoralia, which means 8. And the normal corals you might be familiar with, stony corals of a coral reef, they have 6 or multiples of 6 tentacles, but the octocorals, pretty much all of the corals that we'll see today always have 8 or multiples of 8 tentacles. And this one is already starting to retract, so we won't get a glimpse of the 8 tentacles on this. And it's a relatively small number of polyps. There's only maybe less than 20. It starts off as a single polyp, it then clones off the different other individuals, and other octocorals we hopefully will see later on in the dive can be many, many hundreds of polyps.
Where's this headed after we collect it?
Starboard A or B.
Okay.
Not in
There it goes.
Rock slide.
You got me to push in?
I just tapped a rock and it slid down.
Yeah, you'll have an enemy situation again. Don't want that.
Okay. Yeah, can you zoom in, Jonathan? Shit. There you go.
Bounces back.
Now it's squished. Got it. Need to reset, I think.
Yeah, you're 90 degrees on that.
Yeah, that's what's really stopping me. What am I doing here?
It's full wide.
There we go.
Much better.
Yes. As long as that rock doesn't keep sliding. You can zoom a little bit more, Jonathan. Thanks.
Has the rock been sort of crumbly like this all throughout the area?
Sorry, Megan, we didn't quite hear that.
I was asking if you're seeing this kind of crumbly, breakable rock throughout the area.
We've seen a few different kinds of rocks on this particular dive, so it looks like there's a mixture of geology going on. We picked up a piece of quartz earlier, pure white, that had an encrusting black surface, so that was a bit of a surprise. Whereas earlier in the dive we did get some other rocks with an iron manganese crust that is one of our goals. We really can't tell what kind of rock this is, it's so covered in sediment. But it's a bit of a slope, and we did see a lot of instability in the rocks we were collecting earlier. So there's a lot of loose rocks basically on this slope that we're seeing.
Nice.
Oh, nope.
That'll be what you need to get it.
You think so?
I think so.
Yeah, it's kind of small.
There you go.
There we go.
Oh, well done.
Nice grab.
Perfect.
Well done, Jake.
Thank you.
Coming wide?
Yeah, don't speak too soon.
It'll still give up its chemistry.
Yeah, there we go.
But we never count a sample until it's in the box.
Yeah. We'll have to see if this one's floaty.
So there's a couple of spaces.
Oh, there it goes.
Yeah.
Whee!
Well, now it's shrunk so much, maybe the slope will pick it up.
Good point.
Let's
If
zoom
it
out.
hasn't fallen down the slope.
Yeah, I think if I